I'm Ashley Chang. And I'm Elizabeth Lee, and this is our protein synthesis video project. Protein synthesis is the process of building proteins, and it occurs in two main stages, transcription and translation. Transcription takes place in the nucleus of the eukaryotic cell, where DNA is stored. The genetic code, which is a set of instructions in a gene that determines how to make a protein, is used to transcribe DNA into RNA. So at the initiation site, helicase unwinds the DNA. Then during elongation, the RNA polymerase moves along the template strand of the DNA to synthesize a single-stranded RNA transcript. So messenger RNA nucleotides are added, which are adenine, uracil, guanin, and cytosine. They are added in a 5' to 3' direction that is antiparallel to the 3' to 5' template strand, following the direction of transcription. The nucleotides are added one by one, however, we glued them together beforehand to save time in the video. mRNA is the genetic material that tells the body how to make proteins. Transcription ends at the termination site where RNA polymerase reaches a base sequence that tells it to stop. The RNA transcript goes through mRNA processing, which is where introns or non-coding parts are removed, and exons are put in to replace the introns. A cap is added to the 5' end to protect the mRNA from degradation and initiate translation, and a tail is added to the 3' end in order to stop degradation and help with the export of mRNA to the cytoplasm.
In order to reach the cytoplasm from the nucleus, the mRNA is transported past the nuclear membrane through nuclear pores and into the cytoplasm. Translation occurs in the cytoplasm and begins when a ribosome that is composed of small and large subunits attaches to an mRNA molecule. In cytoplasm, there are many tRNA molecules available, also known as transfer RNA, that carry an amino acid, the monomer of a polypeptide, and an anticodon. There are 3 sites in the ribosome that the mRNA shifts through, the A, P, and E sites. Translation begins at the start codon AUG, which is not shown here, but we'll say the first 3 will be the start codon, and moves in a 5 to 3' direction. In the A site, the ribosome reads the mRNA code 3 bases called codons at a time and adds the complementary anticodon found on 1A of the tRNA. The anticodons provide the information that determines the attached amino acid. Then, with the first tRNA and amino acid in the P site, so we'll move it to the P site. The next tRNA with its own amino acid will move in and attach its anticodon to the complementary codon on the mRNA transcript. The 2 amino acids will link together by peptide bonds, which is represented by the glue. Then the first tRNA and its anticodon will detach as it enters the E site, and then it is released, leaving behind the amino acid. This process will continue and another peptide bond will be formed. Okay, so this process will continue until the stop codon, which is either UAA, UAG, or UGA, is read, and the amino acid chain detaches, and the polypeptide chain is formed. It is released out of the ribosome, finally resulting in a fully functional protein.